Okay, coming to biochemical test. So this table is showing name of various tests and the detection the findings which we get in the urine. So Benedict's test give you presence of sugar or reducing sugar. We can say next is Rothra's test, that is for ketone bodies. Then Hayes sulfur test, that is for bile salts, and then Faucher's test, that is for bile pigments. Okay. So, coming to LH aldehyde test, aldehyde test, it is for the detection of urobilinogen and prophylinogens. Rutra's test, as told, it is for the ketone bodies. It gives presence of acetone and acetoacetate. It may also give you a false positive test in presence of other ketone bodies as you get in phenylketonuria. Next test is Next test is xanthoproteic test, that is for the presence of that is showing the presence or absence of a protein, and it gives a dark yellow precipitate for the presence of protein in the by the use of nitric acid. Next is biurate reaction, that gives quantify test for the protein, and the analysis is made by photometric spectrum. Next is histidine loading test, that is for the folic acid deficiency. Then coming to test for cholesterol, the name of the test for cholesterol are Salvosky, Lieberman, Zeeman, and Zwanger's test. Then for the carbohydrates, for carbohydrate, the tests are Molesh for all the sugars, Salvanov for defructose, and Pearson's test for the presence of carbohydrate. Next is orthotoluidine test, that is for the detection of chlorine. That is for the detection of chlorine. Next are the tests for the proteins. So test for the protein first about the color reactions. Then in that first we have xanthoproteic reaction, which uses the nitric acid. In presence of protein, it gives you dark yellow precipitate. It is used for the detection of aromatic amino acids, that is tyrosine, tryptophan, and phenylalanine. Next, for the presence of tyrosine, we use Millen's test. Then for arginine, we use Sakaguchi test. Then for cysteine, we use Sulvian or sulfur test. For cysteine and methionine, we use lead acetate. For histidine, we use Pauli's test. For tryptophan indole we use Hopkins, Cole, and aldehyde reaction. Then coming to the quantitative assay, for the detection of number of amino acids. For all the amino acid, we uses Nin-hydrin reaction. Then for better quantitative assay, we uses fluorascarmine test. Then coming to ammonia detection, we uses Von-Skelke method and mass spectrometry. Coming to peptide bond, we uses biurate test. Then for peptide chain sequencing, we uses Admin and Sanger's reaction in the inner membrane of the mitochondria. Biological oxidation takes place in the inner membrane of the mitochondria. Uh, then about RBC, RBC gets its ATP or its energy from the anaerobic glycolysis, that is earlier told. The cycles which are taking place in the RBCs are pentose phosphate shunt and methemoglobin reductase system. These two cycles are taking place in the RBCs. Next, RBCs lack mitochondria. That's why they do not have any of these cycle which are taking place in the mitochondria. So remember, Krebs cycle are not present in the RBCs, and as there is no nuclei and presence of HLA antigens are there, so there will be different mode of energy consumption in the RBCs. They get most of its energy from the anaerobic glycolysis and produces lactate. Okay, the end product will be lactate in these conditions. So as there is no mitochondria in RBC, so there will be no ATP production in the RBCs. Okay, next are enzymes. So coming to different terms, holoenzyme. Holoenzyme is holoenzyme. It is composed of apoenzyme, that is a protein, and a prosthetic group or non-protein part. So protein part is apoenzyme and non-protein part is coenzyme or called cofactor. So these can be your inorganic ions or can be complex organic ions. So manganese, phosphate, these are all coming under your non-proteinous or prosthetic or cofactor group of the holoenzyme. Next term is cofactor. What is a cofactor? It is an inorganic ion like manganese, phosphate, and they are required in different reactions. Isoenzymes or isozymes, what are these? These are physically distinct versions which are used for catalysis and they are protein catalyst. Example are LDH and CPK. Ab enzymes are antibodies with catalytic activity. Next is ribozymes. These are RNA molecules. They are not proteins, but they act as enzyme with catalytic activities. Zymogens like pepsinogen, trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase. You all know their function in breaking down in the metabolism of different structures. Then there are some earlier asked questions. What is coenzyme Q? It is a non-heme iron sulfur protein present in the mitochondria. Non-heme iron sulfur protein present in the mitochondria. Cytochromes, catalase, peroxidase, hemoglobin are iron porphyrins. Allosteric enzymes are like phosphofructokinase. CYP3A4 by 5 is the most common cytochrome involved in the biotransformation of largest number of drugs. Suicidal enzymes, the name are clooxygenase and xanthine oxidase. What is ribonuclease P? It is a ubiquitous ribonucleoprotein enzyme. Next are hepatic microsomal enzymes. They are located in the smooth endoplasmic reticulum. They helps in the oxidation, reduction, and conjugation reaction. They act as both exogenous and endogenous substances. NADPH and oxygen are required for the oxidative reaction carried out by monooxygenases. These depend upon the catalytic activity of the enzyme system. Coming to the another term in hepatic microsomal enzymes are functional plasma enzymes. Examples are lipoprotein lipase, pseudocholinesterase, protein factors, prothrombin, and proenzyme. They all help in the blood coagulation. These enzymes are very much rich in the liver, richly present in the liver. They are present all the time, and they function as proenzymes in the blood. So this is the list of different enzymes which helps in the clinical diagnosis. SGOT is associated with the diagnosis of myocardial infarction. SGPT is associated with viral hepatitis. Amylase and lipase are associated with acute pancreatitis. Phosphatase acid is associated with metastatic cancer of prostate. Alkaline phosphatase is associated with bone disease and obstructive liver disease. Coming to the centers point, the amino acid at the catalytic site of different enzyme. In hexokinase, histidine is present. In carboxypeptidase, histidine, arginine, and tyrosine is present. These are amino acids which are acting in the catalytic function of these enzymes. So in chymotrypsin, histidine, serine, and asparagine are present. In trypsin, thrombin, histidine, and serine is present. in phosphoglucomutase, ALP, and ACH esterase. Acetylcholine esterase, serine is present. In carbonic anhydrase, cysteine is present. In aldolase, lysine is present. So, the inhibitor of these enzymes are given in this table. For aconitase, trans-aconitate is the inhibitor. For succinate dehydrogenase, malonate is the inhibitor. For alcohol dehydrogenase, ethanol is the inhibitor. For sodium potassium ATPase, digoxin is the inhibitor. For HMG-CoA reductase, HMG is the inhibitor. For carbonic anhydrase, acetazolamide is the inhibitor. For xanthine oxidase, allopurinol is the inhibitor. Coming to the topic proteases. So this table is giving you the table of different enzymes and their role and the name or example of these enzymes. So proteases basically endopeptidase. It cleaves or it breaks the amino acid peptide chain in the middle. The example are trypsin, chymotrypsin, elastase, and thrombin. Exopeptidase cleaves at the terminal. The example are carboxypeptidase A and B, aminopeptidase, and prolidase. Cysteine proteases, uh, example is papain. Aspartyl proteases contains aspartic acid as nucleophilic amino acid. Example is pepsine and renin. Metalloproteases, example is carboxypeptidase. Protease inhibitor used as drug are captopril and ritonavir. Next topic is detoxification or xenobiotics. The main site for the detoxification of drugs is liver. Histamine and nicotinamide are metabolized by methylation. Natural estrogens, epinephrine and norepinephrine are metabolized by O-methylation. Conjugation with D-glucuronic acid is employed for detoxification of bilirubin, benzoic acid. Detoxification of bilirubin, benzoic acid and aromatic acids, phenol, pyridine and chloramphenicol, thyroid hormone and steroid derivatives. Next, about cytochrome P50. It is an inducible enzyme and it's present in the endoplasmic reticulum and liver microsomes. It is chemically a heme protein. Next, about bowel detoxification. Uh, these are war poisons and it contains arsenic, gold, mercury, and cadmium. About free radicals, they contain single pair unpaired electrons and it causes damage to the cells, proteins, and DNA. Cells are normally protected by these from antioxidants and free radical scavengers. About hemoglobin, iron is present in the hemoglobin in the ferrous state and bound to the histine by 4 pyrols. Globin chain here consists of histidine, arginine, and lysine amino acid. His argylli, that is the mnemonic we earlier discussed. Here, each molecule carried 2 iron ions, that is in ferric form. One molecule of hemoglobin binds with the 4 mole of oxygen tetrameric structure. One ferric molecule binds with the 4 pyrrole rings in the structure heme. Each gram of hemoglobin carries 1.34 milliliters of oxygen. About heme, it is a prosthetic group of hemoglobin, myoglobin, and cytochromes. It is tetrapyrrole with the iron. Iron is in the ferrous form and is located in the middle ring. Mature RBCs here does not contain mitochondria, that is earlier told, so no ATP are produced in these RBCs. They use glycolysis, anaerobic glycolysis for metabolism. Enzymes such as catalase, peroxidase also contains heme. Heme synthesis enzyme is ALA synthase, which is the slowest enzyme, and ferrous oxidation product of heme is hemin. Coming to hemoglobin, iron remains in the ferrous form, that is Fe2+. in both oxyhemoglobin and in the reduced hemoglobin. It will remain in the ferrous form, that is Fe2 plus. In methemoglobin or in ferritin or in transferrin, it is present in the ferric form, that is Fe3 plus. Okay. Coming to methemoglobin, it is ineffective in oxygen transport. And coming to hemoglobin electrophoresis topic, basically hemoglobin is negatively charged molecule and will migrate towards the positive side, positively charged anode. Here the technique used in high performance liquid chromatography used in the diagnosis of thalassemia. So the commonly asked questions from the hemoglobin are beyond infancy, RBCs from the patient with HPSS, sickle cell disease, contains 2 to 20% of fetal hemoglobin, and normal level of HbA2 and HbA is notably absent. Adult hemoglobin will be absent in these conditions. In beta chain hemoglobinopathies, like in sickle cell anemia, HbF level will be increased. Fetal hemoglobin will be increased. Level of HbA2 is increased in beta thalassemia trait, megaloblastic anemia, and iron deficiency anemia, and alpha thalassemia. Next important point is hemoglobin makes its appearance in RBC in the intermediate normoblast stage. Nucleus disappear from the RBC in the late normoblast stage in the process of RBC formation during erythro. Coming to the next topic, the level of hemoglobin. So, level of early anemia should be between 10 to 11 grams per deciliter, and less than 10 grams per deciliter gives a marked anemia. Normal hemoglobin level in newborn should be 16 to 18, and if newborn is anemic, hemoglobin level will be less than 13 in this sick, and clinically stable newborn it will be less than 8 gram percent. 1 gram of hemoglobin binds with 1.34 milliliters of oxygen. It contains 3.34 milligrams of iron. These are commonly asked questions. Arterial oxygen tension is almost 100% at 90 to 100 mmHg in adult and 70 to 80 in newborn. And these are the products where you will be finding the iron. Next, about the cytochromes, these they are all dehydrogenases except cytooxidase. To porphyria, so these are all inherited as autosomal dominant diseases except CEP. That is because CEP is because of the Europe enzyme involved here is uroporphyrogen type 3 synthase. It is of basically two types, erythropoietic and hepatic. Erythropoietic one has congenital erythropoietic porphyria or protoporphyria. Hepatic one has acute intermittent porphyria, porphyria cutaneous tarda, and hereditary coporphyria and variegates of porphyria. Next is cutaneous porphyria. It includes PCT, that is porphyria cutaneous tarda, CEP, that is congenital erythropoietic porphyria, erythroporphyria, and hepatoerythroporphyria. Okay, so these all include cutaneous manifestations. Then lead inhibits ALA dehydratase and ferrochelatase enzyme. Okay, this table is showing the difference between myoglobin and hemoglobin. So myoglobin binds with 1 mole of oxygen per mole, and hemoglobin binds with 4 mole of oxygen. It is tetrameric. Myoglobin is monomeric. The dissociation curve for myoglobin is rectangular hyperbola, and for hemoglobin it is sigmoid shape. Then the curve is left to the hemoglobin in case of myoglobin. Myoglobin takes oxygen from hemoglobin in the blood, Oxygen affinity in hemoglobin is less than myoglobin. It shows no Bohr effect. Bohr effect is present in the hemoglobin. Hill's coefficient is 1 for myoglobin and 2.8 for hemoglobin.